I'm here to talk to you today about AML and genetic testing. Like we discussed, I wanted to go over the fact that AML is not a common cancer, and it can occur in children, but is most prevalently found in the older adult population and more commonly found in men and not women. And the risk factor for getting it is incredibly low. So typically your doctor is going to want to do genetic testing because you presented with some symptoms that are concerning. And I do have some notes that I'm following along here, so just bear with me. Environmental factors can be a cause of in AML, and typically blood testing, bone biopsies, and then moving on to molecular and genetic testing is required. Based on information from the National Cancer Institute, there's a 1.1% prevalence rate of AML for all cancers in the U.S., and an estimated 20,800 people will have AML in 2024. There is an estimated 5-year survival rate of approximately 31.9%, but that does not mean that you necessarily fall in that category. Some of the risk factors that are a cause of AML or are correlated with AML are, like I said, being male, having a familial history of AML, or having received chemotherapy or radiation therapy within a 7-year period prior to presentation of symptoms. Some of the things we're going to talk about are DNA methylation and acute myeloid leukemia. When DNA methyltransferase is inhibited, which this is an enzyme, the methylation or the addition of methyl groups to the DNA can't occur, and this is part of epigenetic changes that occur within the gene. Genetic heterogeneity happens when there's a single gene that has different mutations, but ultimately results in the same disease. AML has varying subtypes, or you could say different versions of it that occur by modification of specific genes on the genome. Let's see. DNA methylation dysregulation is caused by recurrent mutations, suggests epigenetic plasticity associated with apparent DNA methylation, so this means non-normal changes. Let's see. Tumor suppression mutations of AML are characterized by dynamic genomic instability. They contribute to the onset of progression of AML pathology, and tumor suppression genes are associated with proliferating malignant cells, so vast growing of cells that are detrimental to your health and can move to different areas in the body. One of the processes you're going to have done for testing is DNA sequencing, which is one of the most important tools for biologists and geneticists. It allows us to understand and determine the structure and function of your genes and evaluate them on a molecular level, so the tiniest level possible, so we can see the most minute changes and what's going on inside of you. So when we understand genetic makeup, we're looking at the bases of the sequence of the DNA and the chromosome regions. Sorry, 1 second. Another important thing that happens in DNA sequencing is incorporation of ddNTPs, which are used to stop the elongation process. So when you're running DNA sequencing, there is fluorescent material attached to each of the ddNTPs, and those show up as a certain color. You're able to evaluate them from your genetic material and come up with a base sequence to evaluate and give a complementary strand. Moving on, polymerase chain reaction, or a PCR, is a test used to make copies of DNA fragments, creating a plasmid so you can do more testing. A simple way to describe this to you is looking at a piece of your DNA and trying to take one segment of it. And this segment is what we're going to use to make copies, so we can amplify it, like with a magnifying glass, to be able to see more clearly what is going on with a particular mutation or not. So with PCR testing, we are going to denature, which means to separate DNA fragments, place it in a test tube, and then we're going to heat it to 96 degrees Celsius, which is pretty hot. Once that happens, the separated pieces are going to be cooled down to about 55 degrees Celsius, and then we're going to add a lot of primer to the solution. A primer is something we've specifically decided upon earlier, and we don't make that. It comes from a manufacturer, and we use a genome mapping tool to decide what primer we're going to be searching for with what type of disorder. So after the primer is added, we're going to heat the solution back up to 72 degrees Celsius, and we're using a high heat tolerant primer, actually found from the ocean, and this, along with nucleotides, continues to replicate over and over and over again, which each cycle of each of those temperatures that I mentioned, and it'll double the amount of DNA that we're able to view each time. So after you get past a few cycles, you're starting to get a lot more pieces of the genetic material that you're evaluating. Real-time PCR is a technique that a researcher can use to, in real time, evaluate what's going on with their solution and better able to evaluate the effects and what is going on. Fish DNA sequencing, which this is FISH, F-I-S-H, it's abbreviation for in situ hybridization, and this really just means kind of like pausing or affixing the chromosome material in place so you can evaluate it further. The specific sequences that you're looking at are allowing you to map cytogenically, and a stain is added so you can see the pieces better on the slide. Genetic testing of the term FISH is becoming more widely and commonly used with clinicians because it allows them to determine chromosomal abnormalities and detect changes in chromosomal structure and whether deletion or duplication or other abnormalities have occurred. The next generation sequencing is automation of various types of DNA sequencing and allows for a vast amount of genetic material with fluorescent labels to be evaluated all at once, which is called parallel sequencing. I didn't mention in the FISH sequencing that something called primers are attached to the molecules that will connect to the genetic material within the chromosome. That's evaluated with a special fluorescent, a special microscope that can view the fluorescent material. A good way to explain this would be something that I think of is the very bright fish at the pet store that have fluorescent markers in their genes that makes them glow. That's similar to what we're doing with basically a biomarker and being able to track it, and we can see where it lands within the chromosome and find out where an abnormality or defect or addition might be. The process of karyotyping is using a light microscope to be able to evaluate chromosomes treated with a specific dye, and again, the process of fluorescent microscopy is used to identify cellular structures, which is the chromosomes, and you're able to map the entire number of chromosomes, but this process is a little bit slower and takes up to 3 weeks. In order to do this process, you're going to take a blood sample usually, or bone marrow sample, and you will be able to centrifuge or spin the sample to separate it out, and you'll get rid of the liquid that forms on top of the tube, and you will have the condensed bit of blood and cellular material on the bottom of the tube. And then we're going to add a hypotonic solution, which means a solution that has significantly less solutes, or like solid material, within kind of like a water solution, than what is remaining in the tube. This means that things try to balance out. So when this is put in a hypotonic solution, as a cell is placed in that, the cell is going to absorb water because the solutes that are in the solution or in the cell can't move across their membrane. So this will make the cells larger and have more volume, and they could even burst in some instances. So as you know, your doctor has done a thorough physical exam and medical history, which is really important for determining what you have going on. And we want to know what symptoms you're feeling, when they have started, what has changed, and how you're feeling. And we do this in order to also discuss potential risk factors that may have brought upon AML. But in your case, you have received chemotherapy, so this is potentially a likely cause, which is why we definitely need to do the genetic testing to determine if there is a chromosomal issue and if you do in fact have AML. Patients with AML usually are diagnosed when a 20% or more amounts of myoblasts, which are the cells that make all of the different types of blood cells, are present, and there is a way too few red blood cells or platelets. Another test other than a lab draw for a blood sample would be a bone biopsy will need to be done, which may sound a little scary, but your doctor knows what they're doing, and you will have a numbing agent placed over the area, which if they're doing a bone biopsy will likely be on the back area, back here on your pelvis, or if they're doing an aspiration, they may even do it right here. But you won't be able to feel this because they're going to give you something like lidocaine injection, something that will make you not be able to feel it, and your doctor can discuss with you aftercare procedures. Some of the other procedures that you may need to have done other than the genetic testing may be a spinal tap in some instances, depending on symptoms, which at this point it doesn't seem like you would need one, but it's good to be prepared. This also would not be very painful because you would have a numbing agent, but you would feel a lot of pressure. As with anything, there are some risks, but your doctor can go over any questions or concerns that you may have on that. Sometimes testing might take a while. Cytogenic testing, so like mapping the chromosomes and karyotyping, can take 2 to 3 weeks at minimum. And this is something to keep in mind when you're waiting on results. I would also like to mention that there are some really great support services available through the hospital, and I would like to refer you to the genetic counselor that is right up the hall. And I'd also like to give you this information on an AML patient support group and some support groups for your family to attend as well. To recap on the reason for the genetic testing that we're doing, the PCR type of tests are completed because they're very sensitive and can detect even very small changes in the cells, whether that be detecting an illness or detecting still some leukemia cells after treatment. Karyotyping is used to evaluate genetic abnormalities and determine whether there's been translocation, so movement of where a chromosome should be to a different location. Inversion, so still attached kind of in the right spot, but maybe flipped or reversed. So not technically attached the way it should be. deletion when there's a chromosome missing, or duplication when there's an extra chromosome. And let's see. Like I said, with cytogenic testing, the cells have to be grown for at least 2 weeks in the lab, so that's a lengthy process, but can help determine specifics about what's going on with chromosomal changes. DNA sequencing is used to determine the exact sequence of the DNA and the specific order of its code, thereby understanding if there is any mutation or mistake going on with the code, or an inherited disorder that might be present. Some other tests that I'm not sure I mentioned yet that you may need just to have a more complete picture of your overall health and what's going on would be maybe an X-ray or a CT scan or ultrasound or MRI. If you don't know what those are, we can go over that. But basically, they all are different variations of imaging depending on the area of the body. MRI is based on magnets, which is pretty amazing. Ultrasounds, which you may have seen before, kind of just looks like a little something you might even use on your face. It just is like a little flat thing, and it's able to see through your soft tissues without harming you. And an X-ray takes a picture through your soft tissue so you can see the bones or other structures within the body. So I just want to reiterate that your healthcare team is here for you. If at any point you're feeling uncomfortable with any information that's being given or any tests that have been ordered or procedures that may be coming up, please ask a provider or staff member to clarify or explain in further detail. They're not going to be annoyed. We want you to be comfortable and understand and feel like you're part of the process, because you are part of the healthcare team that is serving you. And like I said, I want to suggest the genetic counselor that's here on site, and also suggest some support groups for you, and give you my information so you can feel free to contact me with any further questions or follow-up concerns you may have. And we can schedule a video chat or in-person one-on-one, or you can bring your family to go over some more questions you might have. I hope that this clarified a lot of your questions and concerns about genetic testing and has made you feel a bit more informed about the entire process. Thanks for coming to meet with me.